thank Pat Hines for inviting us again. I love this 20-minute rapid dating routine. This keeps everyone awake for the rest of the conference. So thank you very much for having us. This is a great opportunity. I think Mr. Gerstenmaier did a really good job this morning of illustrating the importance of ultimately creating an economic model and commercializing low Earth orbit and space. And as many of you know, CASIS was created, in fact, to help with that mission. Specifically, we were put in place as an NGO to enable the utilization of the International Space Station, quite frankly, one of the largest capabilities that currently exist in low Earth orbit, and to make that station, that laboratory, if you will, available as a national lab. So that for the first time ever, if there was an interest in doing research, not necessarily from the perspective of doing space-based research for the purpose of enabling opportunities in space, but rather doing space-based research from the perspective of enabling opportunities that can be benefited here on the ground from a terrestrial perspective, the national lab notion, the model that was put in place back in 2011 through the 2010 Authorization Act is what really created that opportunity for the first time. And so CASIS has been doing that. We celebrated our third birthday just last month. And at the end of this past fiscal year of 2014, I'm happy to say that we've enabled over 61 research opportunities on the ISS by supporting through seed and incentivization approximately $14 million. What's interesting, while the enabling of the ISS's utilization model from a national lab perspective, and then sort of coupling with that or adding on to that, the notion of targeting the commercialization of the ISS in addition, our portfolio that I speak of has supported over 67% commercial interest. What's also interesting is that among that portfolio, greater than half, approximately 54% of our portfolio is targeted toward biomedical research. And so I felt it would be relevant today to bring in Dr. Paul Reichert, who is a principal investigator. He is our customer. He supports research that's being fully funded by Merck Research Laboratories out of New Jersey. What's really interesting is that Paul has quite a robust pedigree and a history in spaceflight experience. In fact, 11 missions were conducted on the Space Shuttle program, looking at various interests throughout their R&D portfolio. But with that, I want to introduce Paul and ask him to speak for a few moments on the research that was enabled by ISS, funded by Merck, and where he is with that, what he intends to get out of that research, and hopefully what the near future will hold. So Paul, thank you.
Thank you, Duane, and I'd like to thank Pat for giving me this opportunity to talk to you. For me, this is such a great experience. It gets me out of the lab. I tell people I like to dedicate 50% of my time in the lab, so getting away and getting to do a meeting like this is extremely exciting. What I want to talk to you about today is basically how I hooked up with CASIS, what my experience has been, why Merck is so interested in doing microgravity research, and give you some idea of where we see opportunities of using microgravity in a diverse number of different processes. In 2003, after I had done my last shuttle experiment, I never thought I'd have another opportunity to do microgravity research again. And then in 2012, the chief scientific officer, Dr. Tim Yates-Man, solicited proposals for doing protein crystallization experiments. CASIS had already identified protein crystallization as an area which could benefit from microgravity. The proposal had to emphasize that it had a benefit to human health. The cost would be shared payment in kind, and that it required minimum astronaut intervention. We had to use existing flight hardware, and we both agreed that it's very important to have true ground control experiments. Surprisingly, we very quickly, between NASA, CASIS, and Merck, were able to establish a legal agreement where the emphasis was on publishing and not worrying about the IP position. So in a matter of a month, we were able to draw up a legal agreement, and that made everybody happy. And we all knew that this would involve a peer-reviewed proposal, review selection of the experiments that would actually fly. So what do we use these protein crystals? You know, I think over the NASA early program, there was a lot of work being done on protein crystal growth. And primarily, the goal of protein crystal growth at that time was for structure determination. And the reason why these crystals, protein crystals, are so important to the pharmaceutical industry is that if you can see the three-dimensional structure, not necessarily of the protein, but really what we want to see is the small molecules that bind and either inhibit or enhance the activity of that protein, that those small molecules can be the basis that medicinal chemistry teams can then use to develop druggable compounds. Compounds that have the characteristics where they're potent, they're safe, and they're stable, and they give the most benefit to our patients. And I would say that up until now, that has been the major emphasis of the pharmaceutical industry. But the pharmaceutical industry is changing, just like the aerospace industry. We now consider ourselves a biopharmaceutical company. And 40 to 50% of our research is devoted to biologics. Biologics being monoclonal antibodies and vaccines. And these new biologics require new product developments. They require new means to deliver them to our patients. One of the big issues with monoclonal antibodies is that they're not very soluble, and they have to be given in great quantities. So a typical dose for a monoclonal antibody requires a constant infusion over 4 to 6 hours in a hospital setting. It's not a very desirable way to deliver a drug. So there's a lot of different people looking at different ways to come up with a highly concentrated formulation for these biologics. And one of them is the idea that if you can make a concentrated crystalline suspension that you give as a single sub-Q injection in a doctor's office would have not only great marketing advantage to your company, but also would help in the compliance and the overall benefit to our patients. And the other opportunity that we see is in pulmonary delivery. If you can come up with particles that are about a micron in size and produce an aerosol with these, and you inhale these particles, they're delivered to what's called the pulmonary area of your lung. And you cannot expel these particles. So you can get systemic delivery of your drug through this pulmonary section of your lungs. There is also opportunities in manufacturing. It just so happens that in the manufacture of these monoclonal antibodies, there's multiple very expensive chromatographic steps. And one method that's been used for 50, 60 years to purify proteins has been simple crystallization processes. So people are revisiting the idea of doing large-scale purification using crystallization methods. Also, usually what happens is that a plant will be making this bulk active monoclonal antibody. It'll be storing it, and then there'll be a separate facility. It may be in another country, anywhere in the globe, where then that material will be formulated into your final product. And you need some way of maintaining that active ingredient so it doesn't degrade, but from the time that you made it to the time that you actually formulate it. So these are all things that we're interested in. So then, why microgravity? Why are we willing to put our time and effort into growing these crystals in microgravity? And I think one of the best illustrations of this is in the upper panel of the photomicrograph. This is a crystal, salt crystal actually, that's growing on the ground. And as everybody's seen pictures of astronauts twirling pens and objects in their hands, these pens stay suspended in space. Well, that's the same thing that happens to these crystals. These crystals have mass. And they sediment through the solution, and that causes turbulence around the crystal, and it causes imperfections, and the quality of the crystal degrades. However, if you take that same crystal and grow it in microgravity, you can see in the panel below, you get this beautiful array of dense liquid around the crystal, and the crystal grows in an orderly fashion, much larger. So from a physics point of view, we hope that we reduce sedimentation. Another, I think, opportunity that microgravity has to offer us and is underutilized is the minimum convection currents. A lot of these crystallization processes use temperature to induce crystallization. So if you can get a uniform temperature gradient, you have a very fine means of controlling nucleation and growth of these crystals. And then the final thing is you are able to reduce molecular diffusion rates. And the combination of these physical effects allows you, hopefully, to get larger crystals which have higher internal order and more uniform suspensions. And for us at Merck, it gives us an opportunity to work with entrepreneurs and people in this room. We have to work adapt to the existing hardware and adapt new methods, and gives us an opportunity to work to other state-of-the-art researchers. And let's be honest, it's just cool to do.
So Paul, those physical aspects that you described with the constant temperature, the sedimentation being devoid in microgravity, and the molecular diffusion, those sound like three key and necessary ingredients in your area of work. You can't have one without the other. All three are desirable. Is that something that you can achieve in a ground-based laboratory, or is it necessary to do in space?
I think right now what we're looking at is what benefit do we gain from doing these experiments? And if we do get the benefit, then thinking about in terms how do we come up with ground or earth-based processes that can mimic those effects.
So I'm looking at your next slide here, and clearly there were different perspectives. Every time you conduct an experiment, usually one question that you were seeking to answer generates 12 more. And so the research portfolio continues in a pathway that hopefully you get from fundamental discovery through application. But as we were talking earlier, and I want to kind of go ahead and ask the question while this slide is up, there has been this notion that what is absolutely necessary for the demonstration of ISS as well as low Earth orbit to be successful from an economic perspective and from a commercialization perspective is the sense that we need to have a home run. But I wonder, from what you're describing, it almost sounds as if the ISS as a laboratory, an R&D facility, may or may not create a home run, but what seems to be even more valuable is the fact that it's an opportune way to augment research that can be done on the ground, and perhaps identify ways that will either accelerate or enhance the pathway to market. And I wonder if you feel the same about that.
I think in terms of the opportunity to do this research is having microgravity experiments as a research tool. I think a good example of that would be the U.S. National Synchrotron facilities, where people pay to use access to synchrotron facilities. I see that as a workable model where the U.S. National Lab is used as a unique research environment to do research that you can't do anywhere else. And that will speed up, accelerate the research that you can do. And I think that's more realistic than the thought that you're going to hit that one home run, you're going to cure cancer, you're going to come up with a drug, when right now it takes 10 to 15 years to come up with a new drug at a cost of $1.5 billion. So I think realistically, the idea of little steps and limiting the expectations of these results and expecting an upside from that is the way we look at it at Merck.
So it sounds then that one of the biggest questions we're asked is how long will the ISS be available to us, not only as a physical structure, but as a national laboratory? And we know that we have commitment through the current administration through at least 2024, so about 10 years and hopefully more. That almost gets to this same sort of window of opportunity, if you will, the 12 to 15 years that it takes to go through an R&D cycle to take a product to market. But it sounds as if what you're suggesting is that might not necessarily be the model that we need to look at, but rather having access to this unique capability on this national laboratory when it can be used to augment the research, and then perhaps a larger portion of that work can still be done on the ground, may be a better way to look at this. Is that true?
No, I agree with that. And my hope is that I think when I first started to work with CASIS, I think I was the only commercial person willing to jump in. And then since then, now you've had several other pharmaceutical companies who are interested in doing that. And my hope is that at a certain point, companies will see the value of it and be willing to pay for access to that. So I like the way things are progressing. I see things progressing now.
And that's a good point. So speaking of enticing other commercial interests to ultimately see the value to where they're investing in the opportunity rather than seeking sort of a financial incentive. Just doing the math loosely, you've flown 11 shuttle flights. I'm not real clear on what the mechanisms were that enabled that, but clearly that must have been a $1 billion value proposition that Merck and the earlier form of Merck looked at in order to enable that research. We know that from the work that you've done under CASIS, going forward, there's some other research that probably needs to take place on ISS in order to sort of get to the goal that you're looking for. But in your opinion, your experiences, several opportunities to conduct research, and again, I understand from different aspects, but where do you see this work getting to with respect to ultimately translating into a product in the marketplace?
Well, my hope is that the drug delivery, area, and I think especially the purification potential, are the things that wouldn't require excessive, approval. They could be secondary approvals. Once a drug is approved, the change in the formulation is a relatively abbreviated process. An in-process purification step, could be very simply implemented. So I think there are more realistic short-term, results from that.
Questions from the audience?
Oh, great, thanks. So I'm probably going to hack this up because I can't read the handwriting, but ISS is great for research work, but is there anything in the works for manufacturing? I believe in reference to perhaps the Bigelow module, if that's correct.
10 years ago into the shuttle program, we were dreaming about the opportunity to doing, this in manufacturing in space. And for alpha interferon, which was the product that I was looking at, it was active in a nanogram dosage. It was clear that in a 1 liter bottle, you could produce the amount of interferon that you needed in the globe. So that was possible. I think now with monoclonal antibodies, the dose is 100 to 200 milligrams. So that the size of the processes that you would have to do, it would require a lot of engineering to do that. But it is possible.
Sure. This is a great question. I think I'll take the liberty of answering that. What is the process, if any, when a product that comes out of the significant research, such as what's being done with Merck, is advanced in that accomplishment as spoken for, if you will, on ISS and NASA? How does the ISS and NASA get credit? And how would that be seen by NASA's stakeholders, Congress, the public, as an advancement and a good opportunity? I think there's probably a couple of ways to answer this question. One is that from the CASIS perspective and the research that I mentioned at the outset of this conversation, half of that has had its opportunity to be delivered to the ISS. SpaceX 3 and SpaceX 4 were ones that were really critical to the biomedical research elements. And so as you all in this room know, the SpaceX 3 research was just returned. SpaceX 4 is still on orbit. So we're at this sort of critical breaking point, if you will, to where the results that will come out of this research will hopefully be understood. There is groundwork that needs to be done with respect to understanding the analysis. But from that, I guess to answer the question directly, we feel that we're just now at the point within the CASIS model that we'll be able to reap the benefits of the research that we've supported. And from that, hopefully some of this will be significant advancements in research that we can obviously communicate to a very broad audience and set of stakeholders, not only the general public, but probably equally or more important to our congressional, White House, and NASA interests. So I guess we're good on time, right?
You got 20 seconds to wrap up.
One more question?
Well, thank you,
Okay.
guys.
Thanks, Duane.
Appreciate you being here and talking to us about CASIS and the research that's going on.
Thank you.
Great work.
Thank you.
Great work. Thank you.